Thank you for the introduction, and good afternoon. It's my great pleasure to share some of my work with you all. Today I will talk about the role of bariatric surgery in reducing adipose tissue inflammation and improving vascular function in mice model of type 2 diabetes. As we all know, the prevalence of obesity and diabetes is growing to epidemic proportions worldwide. Obesity is a major risk factor of diabetes. Diabetes is associated with an excessive cardiovascular morbidity and mortality. However, the mechanisms by which the excess fat leads to the impairment of insulin sensitivity and vascular function in diabetes remain to be determined. Recently, a new term called adipose dysfunction has been introduced to describe the pathological changes of adipose tissue during obesity. That is characterized by increased inflammatory cytokine production, inflammatory cell infiltration, as well as the dysregulation of hormones, including the downregulation of so-called beneficial hormones such as adiponectin, and increase in so-called detrimental. So biomarker graph to examine the vascular function. For those who are not very familiar with these techniques, we isolate the mesenteric arteries and mount it in the chamber of organ baths filled with physiological saline and bubbled with oxygen. This mimics the physiological condition. And one side of the case was connected with the force transducer to record the tension change, and the other side was connected with micrometer to adjust the blood vessel diameters, therefore to examine the vascular function. We also used other techniques to examine the adipose tissue inflammation and vascular function changes. We hypothesized that bariatric surgery reduces interferon gamma-mediated adipose tissue inflammation, and the elevation of adipose inflammation contributes to the improvement of vascular function in surgery-treated type 2 diabetic mice. Our surgical model is based on traditional gastric bypass surgery, but was slightly modified, mainly based on the anatomical feature of mice model. Briefly, after the surgery, the food will bypass the upper pouch of the stomach, duodenum, and about 15 centimeters of small intestine, and bypass and enter the distal end of small intestine. We will totally have 7 groups. The control with sham surgery, control mice with 20 days after the surgery, diabetic mice with sham surgery, and diabetic mice 5, 10, and 20, 30 days after surgery. The closed bar indicates the parameters before surgery, while the open bar indicates parameters after surgery. And we can find that the bariatric surgery reduced the body weight to about 15 to 20% in those diabetic mice. It also reduced the abdominal girth, which is a parameter of adipose adiposity. And the reducing adiposity are mainly attributed to the reduction in the fat mass, but not in the lean mass. That's where it's examined by the fax machine. We also found that the surgery reduced mesenteric body weight and mesenteric adipose tissue deposit size. Very dramatically, the surgery reduced the blood glucose level and to the level of equivalent to that of the lean control mice. The surgery also reduced the food intake for about 15%. This is not very dramatic, but it did exert effects on food intake. We then examined the effects of the surgery on adipose tissue inflammation. CD3 is a marker of T lymphocyte. The immunohistochemical staining results shows that the CD3 positive lymphocyte infiltration were higher in the mesenteric adipose of diabetic mice, but were reduced by the surgical treatment. Similarly, the CD3 mRNA expression also higher in the diabetic mice, while surgical reduced CD3 expression. Interferon gamma is an inflammatory cytokine mainly produced by the T lymphocyte. The interferon gamma mRNA and protein expression were higher in the diabetic mice, but were all reduced by surgical treatment. MAX3 is a marker of macrophage. The MAX3 positive macrophage were higher in the sham surgical diabetic mice. The surgery reduced MAX3 positive macrophage infiltration in all the groups. And CD68 is a marker of macrophage, and its mRNA expression will also increase in the diabetic mice and reduced by the surgical treatment. Therefore, the bariatric surgery can reduce mesenteric adipose tissue, lymphocyte, and macrophage infiltration. And also reduce macrophage-induced expression of MCP1. It's an inflammatory chemokine mainly produced by macrophage. The surgery reduced MCP1, mRNA, and protein expression. It also reduced the expression of several other macrophage-derived inflammatory cytokines, including TNF-alpha, MIP-1 and MIP-1 beta. But the surgery didn't affect the expression of several other cytokines that include interleukin-10, MIG, interleukin-6 in retics. Although the adiponectin mRNA expression was not higher in the diabetic mice, the circulating level of adiponectin were indeed reduced in the diabetic mice, and surgery increased the level of adiponectin back to the level of control mice. As I mentioned earlier in the introduction, adiponectin is considered to be a beneficial hormone. It improves insulin sensitivity. Although it's mainly produced by the adipose tissue, with excessive adipose tissue during obesity, the level of adiponectin was actually lower. And that results suggest that the surgery can not only reduce the inflammation, but also regulate hormone levels. So it exerts multiple beneficial effects. So what about the surgical effects on vascular function? We used a myograph to examine the endothelium-independent vessel relaxation of small mesenteric arteries. The results shows that the endothelial function was impaired in the diabetic mice compared with the control mice. 5 days, 10 days, 20 days past the surgery mice shows almost equivalent endothelial function with control mice, suggesting that it really dramatically improved endothelial function. At 30 days past surgery, the endothelial function was only like partially improved, but there were still beneficial effects. This is a busy slide, so let's only focus on the panel D. As I mentioned, the 30 days past surgery only showed partially improved endothelial function. But this improvement in endothelial function was abolished after L-name incubation. And L-name is an inhibitor of endothelial nitric oxide synthase, which means that the impairment in endothelial function in diabetic mice and the improvement of endothelial function by surgery were all attributed to the nitric oxide level, which means the surgery improved endothelial function of those blood vessels by improving nitric oxide bioavailability or production. Those two panels just to show that the endothelial independent vessel relaxation caused by sodium nitroprusside and phenylephrine-induced vessel constriction were not different among all the groups, which suggested that the vascular smooth muscle cell function were not altered. And the surgery also did not affect endothelial function in the control mice. It only exerted effects in diabetic mice. Just to make a little connection between adipose tissue inflammation and vascular dysfunction, We showed that the interferon gamma expression was higher in the diabetic mice, and the sort of function was also impaired in diabetic mice. So what about we treat the diabetic mice with neutralizing antibody to the interferon gamma? Was the endothelial function improved? This result shows that indeed the neutralizing antibody to interferon gamma improved the endothelial function back to the level of the control mice. But if we treat the control mice with recombinant interferon gamma to induce adipose tissue inflammation, the vascular function was impaired. The function is actually equivalent to that of the diabetic mice, which suggested that the interferon gamma-induced adipose inflammation indeed leads to impaired endothelial function. And anti-CP1 treatment also shows similar effects, suggesting that anti-inflammatory treatment can improve vascular function. Well, the treatment did not affect vascular smooth muscle function because the sodium nitroprusside-induced vasodilation was similar among groups. The last slide is just to show that the surgery does not only affect the inflammation and vascular function, it also ameliorates the oxidative stress in both mesenteric adipose tissue and small mesenteric arteries. The superoxide production in the fat and in the small arteries were all higher in the diabetic mice, but were reduced by the surgery treatment. Natural tyrosine is another parameter of oxidative nitric stress. The expression of natural tyrosine was also higher in diabetic mice by those, but was decreased by the surgery treatment. So to sum up, bariatric surgery reduces body weight, abdominal adiposity, and improves glycemic control in type 2 diabetic mice. That's consistent with the results shown in the human being subjects too. Bariatric surgery also ameliorates mesenteric adipose tissue inflammation and oxidative stress and improves small mesenteric artery endothelial function in type 2 diabetes. This is a powerful part of animal experiments because we can easily get the tissue and study different time points. Just some future directions. In this study, we only examined the short-term effects of anti-interferon gamma treatment to make the study more translational. We will need to examine the long-term effects of anti-interferon gamma treatment. And moreover, we should also examine the effects of anti-interferon gamma or bariatric surgery on other vascular beds, because we only examine the effects on mesenteric vascular beds in the study. And the color-restrictive effects of bariatric surgery also should not be neglected. In our study, we only found about 15% reduction in food intake by the surgery treatment, which is sort of low compared with what the surgery can do in human being subjects. But indeed, the glycemic control was significantly improved. So we may need to do the parafed to study the color-restrictive effects. Just some take-home message. Interferon gamma-induced T lymphocyte and macrophage infiltration in inflammatory cytokine expression are the factors of adipose tissue inflammation in type 2 diabetic mice model. In this adipose tissue inflammation can increase the oxidative stress in the blood vessels and reduce the nitric oxide availability in the blood vessels, therefore impairs the function of the blood vessels. And bariatric surgery can inhibit adipose tissue inflammation and improves vascular function in our animal model. I'd like to thank the lab members, several faculty members who provided very valuable input to this project, and our collaborators. Thank you very much.
Got a question? Got a comment?
Yes, I really wonder whether you should call this a type 2 diabetic mouse. It's a DB mouse.
Yes.
And so they have lower activity, so you wouldn't call them a lazy mouse or or an obese mouse. They are a DB mouse. And I think it's misleading to perhaps suggest that all they have is type 2 diabetes. So there are other models of that. The second thing, I'm unclear about the mechanism in your hypothesis as to how bariatric surgery, other than caloric restriction and malabsorption in your bypass surgery, what other mechanism could it possibly be?
You mean in addition to the calorie restrictive effect?
And malabsorption, because you're dissecting your bowel.
Yeah, the only thing I can say is we actually tried the 40% calorie restriction in the same animal model. And although the glycemic control was also improved, but body weight was not changed at all. And that's very interesting to see, because you would assume that the surgery only reduced about 15% food intake, and it exerted dramatic effects on both glycemic control and body weight. But 40% calorie restriction did not affect body weight in those animal models. So there must be something else the bariatric surgery did to affect the conditions of those mouse, in addition to the calorie restrictive effects.
What could that be?
That might be the effects of hormone regulation. Yeah, because the surgery treatment did affect the circulating level of hyperactin, which is a very important hormone, has been published to affect body weight control.
Any other comments? I would answer you basically, we are finding out that the bypass mechanism, by bypassing the duodenal